Okay, welcome everybody. I think we're going to wait just one more minute, let people get signed on, and then I'll go ahead and introduce our speaker for today. Okay, good morning everybody. Thanks for joining us for the IPHC's first seminar of the 2021 series. My name is Ian Stewart, a quantitative scientist at the IPHC, and I have the pleasure today of introducing our speaker, Dr. Tom Helser, who is the Age and Growth Program Director for the REFM Division at the Alaska Fisheries Science Center. I've worked with Tom for, I think, it's close to 20 years now, since he was first willing to take a fresh grad student and pull him in on a few projects and feed him an occasional bowl of cheap teriyaki. Tom has a unique perspective on life history, age, and growth information through his extensive experience and background, ranging everywhere from collecting the data to analyzing life history data sets through Bayesian hierarchical analysis and generalized mixed models, conducting stock assessments from everything from crabs to rockfish to Pacific hake. I think it's pretty exciting that somebody with that kind of experience background now leads a research program developing and using emerging technology to make these analyses more efficient and more informative for fisheries management and for use in stock assessment. So with that, let's let Tom kick off as the first speaker of the IPHC's 2021 seminar series. Go ahead and take it away, Tom.
All right, thank you, Ian. Hello, everybody. Far and wide, I don't actually know who's on this seminar, but I hope that I can give you a good deal of information on the current research that we're working on. From the title, you're probably scratching your head saying, well, can you really age fish at the speed of light? And well, the short answer is no. Not too surprising there, but truth be told, we're employing a technology that has ability to substantially increase the efficiency of aging fish through the interrogation of otoliths using Fourier transform near infrared spectroscopy. And this work has been going on for about a year and a half now. I'll get a little bit more into the details about the genesis of the work. But I have really much of my research team focused on this effort. Of course, being an age and growth and reproductive biology lab, we're very interested in trying to gain insight and efficiencies in basically in getting life history parameters for stock assessments. The whole project began about a year and a half ago, where we started exploring the use of this technology for aging fish by interrogating otoliths. With the success of our first paper that was published in 2019, we submitted a proposal to the NOAA Fisheries Strategic Initiative, which resulted in a multi-year, multi-million-dollar award with which we were able to attempt to operationalize this technology for the enterprise of fish age estimation across NOAA in general. The presentation I'm giving today is going to be organized into multiple parts. The first part, I'll be talking about the background of FT-NIRS technology and how it works, both in terms of the physics behind it and the predictive analytics. Just a note, we are also exploring other means of spectroscopy, such as Raman spectroscopy, to basically ground truth or use an additional tool in parallel to help us understand the relationship between the target properties of what we're scanning with this technology and what we're trying to estimate, such as life history parameters in particular. I'll also talk about the successful applications of FT-NIRS in age estimation. I think we have a number of well-proven papers that have shown the veracity of this technology. We're also looking at using this technology for other life history parameters that are important to stock assessment, such as estimating energy density or the amount of lipids, say, in the muscle and tissue of fish, as well as the reproductive status of fish. I'll also delve a little bit into the application of artificial intelligence and deep machine learning, specifically deep neural networks, to help us take away some of the human subjective aspects of not only the interpretation of the otolith, but also in terms of how we develop these predictive models and update them for future predictive capability. And then finally, if there's time, I'd like to talk a little bit about the strategic initiative pathway and our goal towards the operational readiness within NOAA, within the NOAA enterprise of fish age estimation. So starting it off, FT-NIRS has been used in other technologies for a number of years, specifically, for instance, estimating the fat content in dairy products, the amount of protein in feeds in the agriculture sector, and the active ingredients in pharmaceutical drugs that are used for medicine. Also estimating hydrocarbons and impurities in hydrocarbon and gasolines, and also and more recently in the neurosciences, which is called functional spectroscopy. And then to our thinking, when we first learned about this, is why not employ this actual technology in fisheries biology as a means of rapidly interrogating structures to estimate certain target properties. I do have to admit, however, that ours was not the first paper published on the application towards fisheries biology. Our colleagues in Australia published a paper, I think it was 2017, in which they showed or demonstrated the utility of this technology for estimating the age of shark from vertebrae. So let's now talk about near-infrared spectroscopy and what it is. If you look at the bottom, you can see this is the electromagnetic spectrum of the different wavelengths of energy. And the near-infrared area is right next to the visible range here, and it ranges between 12,500 and 4,000 wave numbers per centimeter. It's in this range here where energy, when it's in contact with substances, causes what are termed molecular vibrations. And molecular vibrations are occurring all the time. It's just that when, for instance, near infrared light, which is targeted on an otolith, which is shown here on the upper left, that has a tendency to cause an excitation from the ground state of energy to a virtual state of energy. And that energy change, it results in what's called molecular vibrations here. And this is shown here with, for instance, hydrogen, which is bonded to different parent atoms. And the types of stretching which are occurring here, say, which are asymmetric and symmetric stretching, and bending, which are asymmetric and symmetric bending with the hydrogen atoms, these have a tendency to be related to specific bonds, whether it's carbon to hydrogen, nitrogen to hydrogen, long chain carbons to hydrogen, and oxygen to hydrogen. So these actual molecular types of stretching or bending relate to a specific amount of wave energy that's being recorded by an instrument, and therefore acts as essentially a fingerprint. Before I get specifically in that, I want to talk about how the molecular motions are detected and measured. Here is a Fourier transform near-infrared spectroscopy spectrometer that we actually have in our laboratory. It consists of a number of different important elements, which are obviously the detector, the interferometer, and the near-infrared source of light. This is from a tungsten globular light source that produces the near infrared energy source. And so what happens is that the instrument is actually measuring these vibrational responses over the time and frequency domain, because we actually we set the parameters for how long we need the instrument to interrogate the subject, which is, for instance, the otolith. And over time, the mirror is actually moving in all different sort of random directions to refocus the energy onto the entire surface of the otolith, thereby not excluding any part of the surface of the otolith to initiate those vibrational responses. So the Fourier transform process of this is actually a transformation to collapse the time domain into just the frequency domain, and that's what we're interested in learning about or identifying with regard to the importance of, say, what frequency response is related to the target structure like the otolith. Ultimately, what we get in the frequency domain is absorbance units that are related to the vibrational response by wave number. This particular this particular absorbance spectrum is actually a fingerprint of the target structure that we just interrogated. And each of these measurements actually occur at a 1 centimeter wave number. So potentially we have from 12,000 to 4,000, or actually 8,000 measurements of the absorbance profile per channel or single wave number. And the fact is that the different ages that are associated with the otoliths produce a different spectrogram, which shows in this case there's an increase, say, both in the absorbance profile and the difference in that absorbance profile as a function of age, which is shown here on the right-hand side of this graph. And so it's in this area where there's different stretching and bending responses where the information content actually exists as it relates to identifying or trying to measure that in relation to the age of the fish. And so you can see here there's a different channels that represent the response function of each of these absorbance profiles. And it's also these absorbance profiles where they have peaks and dips that have a correspondence to the different types of bending and stretching of the vibrational or molecular responses to the carbon and nitrogen bonds. And that's what's important and what we're trying to figure out as it relates to the target properties of the otolith, for instance. So how then do we predict age from otolith spectra? Okay, so if we had a sample of otoliths which come off, say, an observer cruise or a fishery independent survey, what we essentially do is to measure each of these otoliths by the FT-NIR instrument. The scan time is approximately between 30 and 45 seconds. And then for each of these samples, we get an absorbance profile, which is shown here, all stacked up. And we color coded it in regard to the age of the otolith that was associated with that spectral data. And how do we kind of match the spectral data with the age estimates? Is we use the typical microscopic approach to analyze the otoliths using the typical band count method, which may involve also some pre-processing, which is burning or roasting or sectioning. And sometimes actually staining, which can take up to, a number of minutes in the case of, say, walleye pollock, to up to 10 minutes in, say, a vertebrate like skate. But anyways, we can match then each individual age response from the otolith to the response profile shown here. So these are actually a one-to-one correspondence in terms of this individual observation of age and the entire response, which is essentially a multivariate response in terms of the absorbance profile. It's here that we conduct the analytical aspects of the analysis, where, for instance, we may, in the first instance, want to pre-process the data to enhance the signal to noise ratio. a lot of times the pre-processing techniques may involve first derivative transformation or Savitsky-Golay smoothing. And then the second stage is to basically optimize the age estimate with the response or the absorbance profile in this multivariate response. Okay, so in that case, what we're doing is basically a rank reduction of this multivariate response into the dominant eigenvalue and eigenvector pairs, which then produce a predictive function that relates the traditional age with the FT-NIR age that's from the spectra itself. Before we can actually use this in a operational setting, for instance, in an age and growth laboratory, we have to conduct what's called calibration and validation of the model. That's simply basically relating the multivariate response or the rank reduction of that response to the age as a predictor, and then using an out-of-sample data set to validate or evaluate the model skill as it relates to an untested set of data. And what you see up here on the right-hand side is the result of an analysis we did, which I'll show later, with walleye pollock in the Bering Sea. And what this shows is that there's a strong response between the traditional age and the FT-NIR age as estimated through the spectra from the otolith. Once we have a verified model, we can upload that model into the instrument then, and then just go about the activity of taking samples, which are again unknown in age, and in this case, not microscopically aging them, but actually scanning them on the instrument, which I said takes between 30 and 45 seconds, and therefore generating the age compositions that are used in stock assessments. So this is essentially the entire process in the simplified version that shows how you pair the spectra with the otolith. You do calibrate using model predictives and analytics, and then actually generating a predictive model that is used for generating new data product for the stock assessments.
Hey, Tom, we have an online question for you.
Yeah, sure, go ahead.
Yeah, this question's from Andrew Chen, and he's asking, would the profile of protein accumulation be affected by growth?
Yes, it could in fact be. And in fact, that's what we're investigating. That is the amount of protein that's incorporated in the otolith as the animal ages. It may be the primary sort of target material that is responsible for the spectral information that's in the absorbance profiles. This has been not verified yet, but we are doing a parallel set of research where we're conducting some proteomics actually on the otolith material to verify this. So one thing's for sure is that the mathematics can be complex. And one area that we're exploring is the use of machine learning algorithms to basically optimize and reduce not only that multivariate absorbance data, but also to sort through the best regions that are most informative for the prediction of age, as well as the pre-processing optimization tools that would be used to enhancing the signal to noise ratio in that information. And I'll talk a little bit about that later on. So I just want to kind of talk about the veracity or the verity now of the data products that we've generated to date that underpins really the going forward and the utility of operationalizing this technology. In 2019, we held a workshop that included over 100 members of the international national community. And this workshop kind of spearheaded the strategic initiative and served as a source document that identified the operational tools as well as the challenges that we might be encountering in trying to operationalize this or make it operationally ready in the NOAA aging enterprise. This was our 2019 paper published in the Canadian Journal. We also published a paper in the Journal of Near Infrared Spectroscopy that dealt with using spectra from otoliths actually as a means of classifying fish species from different ecosystems. Another paper was published in Proceedings of the 14th International Conference. There was another paper we published as it related to age estimation using NIRS in the ICES Journal of Marine Science. This dealt with another species of fish that was much longer lived than the walleye pollock in the Canadian Journal. And finally, we have another paper that should be coming out soon dealing with another age estimation application for Pacific cod in the Bering Sea. I just mentioned these products because I think that before it's feasible to move forward, we have to demonstrate that the scientific soundness of the utility and the utility of the technology for the interested application that we're trying to solve here. So I'd like to just go over some of the data that we generated from the case study of walleye pollock in the Bering Sea. This was from 2016 and 17 catches. These were survey catches of walleye pollock from the southeast to the northwest Bering, which are the typical bottom trawl survey areas used in the stock assessment. Again, I talked about, not only collecting spectra, we collected something like 3,500 spectra from individual otoliths. We went through the process of pre-processing and generating a calibration model, holding data back, and then doing an external validation. So this essentially shows those results. This is the calibration relationship between the traditional microscopic age, and this is the age generated from the spectra using FT-NIRS analysis. And so what you'll see here is that 89% of the variation in the external validation data of the traditional age was explained by the FT-NIR spectra from the otoliths. We also see the root mean square error prediction of about 0.96, which indicates that 67% of the time we will predict the age relative to the true age or the traditional age plus or minus 1 year. This is the root mean square, or sort of the residual prediction deviation. It's another metric that's typically used in the chemometrics literature. Those that are above 0.25 are generally thought to as being good, but above 3, they're thought to be very good. And so then the question is, how does the two different aging methods compare in terms of precision or readability? Well, this graph shows this precisely, where on the left-hand side, we see the traditional microscopic aging approach, where we generally plot age reader 1 on age reader 2. These are two independent trials of the same otolith. And as you can see here, the traditional metrics that are used to compare these is the percent error agreement. So 67% of the time, age reader 1 and age reader 2 agreed, plus or minus 0 years. CV was basically at 4%, which is pretty good. Then we can see here on the right-hand side, we have individual separate trials or independent trials of the near-infrared spectroscopy. And when we compare those to the traditional age, using basically the ages from the traditional approach and 3 different independent trials, we can see that the percent error is actually 82% in the FT-NIRS method, and the CV is much lower at 3%. This indicates that precision is markedly better than the traditional aging approach. It's probably related to a variety of things, obviously, in terms of the ambiguity of the age pattern that the age readers are seeing. It could kind of remove the prospect of age reader fatigue, but also it's measuring a target property in the otolith that is much more consistently determined from the spectra that's collected from the instrument. Now I want to shift gears a little bit and talk about another age application with red snapper in the Gulf of Mexico. This is a much longer lived fish than, say, Pacific cod or walleye pollock. The top graph here shows the absorbance profiles of a number of different otoliths that were measured from red snapper. And then, of course, what this shows is the pre-processing technique where we're sort of trying to isolate those channels or bands within the electromagnetic NIRS spectrum that is most informative. And in this case, basically, this is a first derivative transformation in a Savitsi-Golay smoothing, which is used to enhance the signal-to-noise ratio. The results of that analysis, again, as we showed before for walleye pollock, showed the relationship between the traditional microscopic age and the FT-NIR age calibration on the left-hand side and the external validation on the right-hand side. And again, in this case, 92% of the variation in the traditional age data was explained by the spectra from the otoliths. And we get a significantly larger RPD and a root mean square error prediction about 1.33. Again, meaning 67% of the time, the age will be within the predicted age, you know, basically plus or minus 1.3 years for an animal that can live to be 4 decades old. And finally, I just want to illustrate a recent work we've conducted on Pacific cod. Again, this analysis was conducted from survey data over a good number of years. I think, again, we had quite a few spectra. In this case, probably more like 4,000 individual otolith ages and spectral absorbance profiles. Again, you know, most of the information in the lower or the upper wave numbers is pretty non-informative. That has to do with a baseline shift in the spectra. And again, this shows the pre-processing. And then finally, what's kind of interesting here is the principal components analysis showing the first and second domains of that multivariate data. Clearly shows that there's a cluster effect as a function of age, from age 1 being highly separated to age 11 or 12 being less separated than the younger ages. And in this case, the results are slightly less, what's the word? Slightly less interesting, I guess, in this case, although we can see that the external validation more or less gives roughly the same result in terms of the root or the coefficient of determination. So in all of these cases of age estimation, we found that the coefficient of determination of prediction was greater than 88%. In some cases, it can be improved with some other techniques that I'll show in a little bit. I'd like to move on to species classification from otolith spectra using diffuse reflectance. In this study, we looked at spectra that was collected from 5 different marine ecosystems, ranging from the New England area in the Gulf of Maine all the way to the Bering Sea. We have a mix of flatfish, groundfish, rockfish, coastal pelagics. Some of the oldest fish, I think, ranged in about 80 years of age. But anyways, again, here's the spectra from the fish categorized by the individual ecosystems, if you will. And we found that there was strong classification accuracy from fish from different ecosystems and also from species within some ecosystems. So for instance, here, like in the Bering Sea, we found strong separation between Pacific cod and walleye pollock, just based on the spectra from their otoliths. I'd like to talk about another example here as well. Alaska has a pretty big mixed fishery catch with both black spotted and rough-eye rockfish that are generally, up until recently, not separated on the deck, and were simply categorized and assessed as a mixed rockfish group. One of the topics we were interested in exploring is whether or not we could use spectra from these otoliths from these animals and to basically attempt to get classification models that could actually identify whether an animal is basically one or the other species from a mixed catch using diffuse reflectance from the spectra. In this case, we can see that this is the spectra, and there's reasonably strong separation between rough eye and black spotted, rough eye being in pink, black spotted being in blue. And this shows a couple of PCAs where you've ranked PC1 on PC2. This, I believe, is the calibration model, and then this is the external validation. In each of these cases, you can see strong clustering and separation between the two species in this ordinal space. And also, when you conduct a cross-classification analysis using partial least squares discriminant analysis or vector machine discriminant analysis or KNN clustering, you can see, and this is based on the this sample here, which is not entirely large, but we can see that the prediction or the classification accuracy is really quite outstanding. This is important because this allows us to go back and just with the scan of the otolith, which takes, as I mentioned, between 30 and 45 seconds, we could potentially classify all historic catches and conduct stock assessments specifically on a species-specific basis.
Hey, Tom.
Yes, sir.
All right, I got a couple questions for you. One is related to this slide in particular. This question comes from Jimmy and Ellie, and he's wondering if those are from similar size or similar aged rough eye black spotted rockfish.
Yes, there is a strong overlap in the age of the rockfish. These, in fact, were a sample of. This sample came from 2019. There were about 1,000 samples. They were genetically vouchered, so we actually know the ground truth of the sample. The age was by and large overlapping to a good degree. I can't specifically speak about the sizes or the weights of the animal, but yes, I would say that the ages were quite a bit overlapping. And
okay,
this is basically a random draw, so it doesn't necessarily, isolate one age group from one species or the other.
Sounds good. And then one other question that might be appropriate right now, and then there's a couple that we can do at the end, but this one's from Catherine Matson, and she's wondering, how does this technique do with crystallized or dysmorphic otoliths, and also how does it deal with plus growth on the edges?
Right, okay, so we do not scan the diagenetic otoliths, those that are, funky, let's just say. We have been scanning some of them to evaluate whether the different crystalline structure in terms of aragonite versus, say, calcite in the carbonate has an effect on the ability to use it for not only aging, but say differentiation in terms of species. So we have not analyzed that data. So I can't specifically speak towards whether or not that's going to be, those types of otoliths will be useful. Plus group is something, or I guess in terms of this, the spectra is actually coming from the entire otolith sample. So age does not enter into this particular analysis like it would, say, in trying to identify the edge type or the plus group for age estimation. Now, that may be more of an issue related to, say, using this technology to estimate the age of fish. Because by and large, there can be a mismatch between, say, the amount of spectra that's collected from an otolith when you scan the entire otolith versus how an analyst identifies, say, the plus group, or whether there is actually much growth at the margin of the otolith. So there is sometimes poor or lack of fit as it relates to the data when that may be ages from from otoliths that have been an age added or something like that. Should I go on?
Sounds good.
All
yep,
right.
go ahead.
Yeah, I can answer more of that too. So the another application I want to talk about is energy density. That is basically predicting, say, in this case, juvenile cod % lipids. The reason this is important is because We know that there may be a fatty acid content in the livers or the muscle of the fish, whereby if the threshold gets below that, the animal may be subject to imminent death. So the goal of this would be to rapidly identify poor condition fish that might lead us to maybe deal with or implement some other management criteria when a population is below condition, like maybe which happened in the marine heat wave, say, of 2016. So this was data collected from a laboratory study from our colleagues up in Alaska in Auk Bay Labs, where they had different temperature treatments and they fed the fish different rations. And what they were trying to do is basically starve some fish or increase or decrease the condition of fish based on the temperature and the feeding conditions, as it would emulate, say, a marine heat wave and the resulting poor condition of fish thereafter. So what we had done is we took the samples that they use for bomb colorimetry and other proximate analysis of percent lipid, and we scanned the samples using the FT-NIRS. And this graph shows a spectral. This is a pre-processed spectral information, again using Savitsky-Golay smoothing and a first derivative transformation. And the color coded is basically going from a low percent lipid from 2% up to 8% from the laboratory studies. And what you'll see here is that there's a lot of information in various regions of this absorbance profile, particularly in the upper or the lower wave number regions between 6,000 and 4,000, where we see the lipid and the protein regions that respond to the near infrared energy. This is the combination region, but there's also overtone regions that also reflect lipid and protein responses. And so it's these that we focused on with regard to developing a predictive model. This graph shows a PCA that's conducted, and here it's plotted in ordination space of the PC1 and PC2. You can see that there's a strong relationship between not only the size of the fish, where fish that were smaller are in the lower corner quadrant here, but fish that are larger and more are lipid rich were in the higher quadrant here. That indicates that there's some response that's being informed by the information generated from the spectral information in the otolith. So when we conduct a predictive model using PLS regression, we can see that roughly 68 or 67% of the variation in the measured lipid content from the chemical tests were explained by the spectra from the same homogenates that were used. So we can plot those as the measured lipid response to the predicted lipid response. And that's a pretty good relationship. We certainly want to increase the sample sizes, and then there's opportunities to add other variates in here that might be better adept in addition to the percent lipid or the spectra in explaining the response. So one thing this says to me is that it has the potential for a rapid field-based assay. I think we need more laboratory studies and chemical ground truthing of exactly what's being measured in terms of the spectral information as it relates to the lipid content in the whole body homogenates. And this is another figure that shows another look at spectral data, but this was actually collected using a Raman spectrometer. This actually is to the right of the electromagnetic spectrum of the near infrared. It's in the infrared range, and it's called the Raman shift. And basically what this shows, and it shows these blocks that relate to the CH2 bonds, whether they're scissoring and twisting in terms of the molecular vibrations, or whether they're CH3s that are scissoring and twisting, and the CH stretching. These are skeletal optical modes. And in essence, what this represents, or it tells us something about what we've seen in the near infrared perspective, is that the lipids are actually responding to hydrocarbon chains that are observed in the regions here that relate to the 1400, 1,300, 1,200, and 1,200 to 1,000 fingerprint regions. So this is actually another parallel analysis that gives us more specificity about the regions and the types of molecular responses that we're seeing in the substance or in the whole body homogenates. I just want to show one more example before I get into the next topic. And this is actually using this information to do rapid analysis of fish reproduction. And we're looking here at walleye pollock. Everybody knows that the reproductive status is important in stock assessments, whether it's generating a maturity ogive or the age at first maturity. This shows the gonad tissue and the histological analysis related to that tissue. One thing to be cognizant of is that detailed histological analysis requires a lot of time. You have to fix the samples in formalin, you have to wash them, you have to basically put them in cassettes and then send them off to a histological laboratory and then read the slides for the histological state. The goal here was to see whether this technology had the ability to actually differentiate between immature and mature fish by scanning the ovary tissue from walleye pollock. And so what we see here is, again, the pre-processed waveband information from the spectra that were collected from a number of different maturity state fish. Again, this is pre-processed spectra. One area here that's particularly interesting as it relates to the lipoproteins and phosphoproteins that are in the ovary tissues and also in the eggs in terms of the amount of that's present in mature versus immature fish. We conducted a partially squares discriminant analysis, calibration, and validation. This shows here the principal components analysis of the latent variables in bivariate space. This is actually with the validation samples and showing clear and discrete separation between the mature and immature fish. Again, we don't have a lot of samples and we're working feverishly to get more samples scanned. But what you can see here is that the prediction accuracy is pretty outstanding in terms of the spectra of the ovary tissue's ability to differentiate between mature and immature fish. And again, this is more or less preliminary because of the low sample numbers, but this certainly gives us hope and energy to basically dedicate more sample effort into identifying this as a tool, this technology as a tool for this specific application. I'm going to jump away from here because what I want to do, this is just another application to northern rockfish, which shows good discrimination as well. One of the things I wanted to jump in is using artificial intelligence and machine learning as a tool to help us better generate predictive models using more variables than simply the spectral data that we're collecting. We had the opportunity and we're fortunate to be involved in a an artificial intelligence project that's spun up by NOAA headquarters. This has given us access to the Google Cloud computing platform, as well as the artificial intelligence tools that are being used in that platform. And what we did here is we wanted to see if deep machine learning or deep neural networks were basically better at or could actually help us generate, predictive models. Because as I said before, there's some mathematics involved where not only are you pre-processing the data to enhance the signal to noise, but you're also trying to optimize the relationship between the target properties such as age and the target substance in the spectral data that you're collecting from that from the otolith, for instance. And so what we employed here was a number of different AI platforms in the Google Cloud computing library that allowed us to generate a number of different models. Okay, so what we did is we took all the data that we had initially published in the CJ FAST paper shown here, where this is from 2016 and 17 data, roughly 3,500 age data and spectral information. You can see that we had an R-square of about 89%. With the deep neural networks, what we did is we generated different models that utilize different tuning platforms, such as hyperband tuning, Bayesian optimization tuning, and then we coupled these, or using both unprocessed and first derivative pre-processing techniques, as well as basic random forest models. But what you'll see here is comparing, say, these models relative to the paper that we originally published with the same data. We can say we can see that we got a little bit an improvement in the R-square up to 0.91, but in fact we actually found a much better improvement in the root mean square error of prediction. That is by decrease in that metric by 17%, which may be significant when it comes to prediction of ages, and particularly where prediction of ages for older age fish might be actually less robust. I want to talk a little bit about how this was set up. So in this case, not only did we use the spectra from the original work, but we also added a number of different attributes that created a 3-layer deep neural network. So we incorporated not only spectral data to estimate fish age, but we incorporated spectral data and otolith weight and fish length and fish sex and latitude and longitude. So what we're doing is basically taking these other attributes and then putting them into the hidden layers of the deep neural network and allowing the machine learning algorithm to sort out the most meaningful and connected interrelationships between these different layers. And a deep neural network is comprised of a number of features, one of which is the visible input layer, obviously, and the visible output layer. But everything else in here consists of a transfer function that basically allows the movement of one solution from the neural network to move forward into the next layer. It involves a set of parameters or coefficients of those parameters, and then some sort of structured hyperparameter set of data that's either sort of based on Bayesian optimization or hyperband, which is more of an orthogonal relationship between the hyperparameters, and it actually forces more of a search algorithm in spaces where there's greater orthogonality in the parameter space. And so this actually shows some of the outputs of those deep neural network algorithms. This basically shows the loss function of both the train and the test set. And so this actually moves forward through these different trials or epochs, where the epoch represents the full forward solution moving through the deep neural network all the way to the end, and then does a backward hindcast and does it all over again for up to, say, in this case, 130 epochs. This shows the error distribution of our final solution. And this basically shows the relationship between the traditional age and the FT-NIR age from that deep neural network. Here we can see that the R-square in this case is a 0.91 and a pretty good reduction in the root mean square error prediction. What I find kind of interesting is the impact factor. It's how these factors are gauged as in terms of how important they are to the final solution. And otolith weight, interestingly, was pretty high, but what I find even more interesting was that the wave numbers in the 4,000, 5,000, 6,000 wave number regions were quite a bit more informative than otolith weight. And again, it's these areas where we generally see a lot of information content in spectra as it relates to the otolith structures. Ian, how much time do I got? I wanted to talk a little bit about the strategic initiative, but if I'm running out of time, then I would defer.
Tom, I think you could go a few more minutes. Go ahead and tell us about it.
Sure. So as I said, NOAA Fisheries funded this 5-year strategic initiative. Obviously, our goal is to try to operationalize or get some operational readiness for this technology for integration, or I should say for generating data products for integration into stock assessments. The actual strategic initiative development team consists of all the 7 primary aging laboratories in NOAA around the country, with the Alaska Fisheries Science Center more or less spearheading this effort and then disseminating technology and data information to the other laboratories around the country. At current, we have a number of spectrometers fielded across the nation. We currently have 2 at AFSC, both the MPA and the Tango. The Tango we've made field ready, where we can actually bring it shipboard and try to get shipboard measurements of age right on the deck of a ship. We also have a high throughput analyzer, which has a sample wheel on it, and we can get up to 33 measurements in an automated fashion. Panama City also has one of those. We fielded 2 more Tangos at the Santa Cruz lab and the Beaufort lab. The goal obviously is region specific because each region has different species and different management systems. So what we've done as a nation is choose 3 different species that we want to attempt to basically proof of concept and apply the technology to, and then move forward into integration into stock assessments. Actually, walleye pollock is high on the list just for Jimmy and Ellie. But we're doing actually 4 of them, in this whole process. So, but anyways, it's important, I think, that we demonstrate the utility across a number of species. It may not be the appropriate technology for all species, particularly species where you could basically take an otolith, put it up to the light and count the otoliths that way, or the otolith or age from the bands that way. Which I don't know many species you can do that, but obviously, yeah, one has to take into account the efficiency gains. We communicate through what's called a virtual lab. We have a number of projects that we're coordinating to answer important questions, including the proteomics of otoliths. These projects are ongoing. We also have a document library in the virtual lab. We share results from workshops and other discoveries on that. The workflow of the strategic initiative beginning in 2020, which was the first year of real funding, started this year during the pandemic, which unfortunately has been somewhat hampered. It's during this time that we wanted to basically, optimize instrumentation, which we actually have done quite well. And we've generated this proceedings that documents a lot of what we've done already, a lot of the questions that need to be answered, the research activities we need to answer going forward. Otolith scanning has been mostly done in our lab because of the delay in fielding instruments to the other labs. I think we're up to about 35,000 scanned otoliths at this point. Also during this phase, the 2021 phase or the application development phase, we're looking to develop predictive models, refine those models, and then actually look at the optimal software and analytical tools to do those calibration validations and then fully implement them into the application implementation phase. It's in this phase over 2021-22, we're conducting simulation studies to evaluate how you detect faults in models, how to do process control. In other words, if a model needs updating, how best to update the models, what are sample size required to update models, how many otoliths do we need to actually microscopically age on an annual basis for updating and that sort of thing. Another area and that's basically we're developing a simulation framework to answer those questions. Ultimately we'll go into a manual of best practices and standards. And again, we're employing topics in machine learning to see whether or not that would be a useful platform to generate these models. The final stage between 22 and 24 is actually the integration into stock assessments of these new data products. The goal here is to evaluate the assessment model outputs to both the FT-NIRS data relative to the traditional data, because the stock assessments currently have been based on the microscopic aging paradigm, and what we are introducing here is potentially a new paradigm of age estimation, and therefore needs to be completely evaluated in terms of the model output. We are also conducting simulations for this as well to identify the benchmarks and standards by which to evaluate and the assessment model benchmarks or metrics, and what types of outputs or in fact inputs are pulling against, say, the new data, or the new data are influencing most substantially. Obviously, we would want to provide the FT-NIRS precision and reliability metrics for, again, use in age error matrices, which are typically integrated into stock assessments. And then finally, how best, if we actually move forward, how best to convert historical age to FT-NIR age, or in essence, how to let the model deal with a new set of age data in, say, specific assessment situations. And so obviously there's challenges going forward. Again, we're conducting proteomics to identify explicitly what in the otoliths that is related to the spectra that we're measuring from the otoliths. We're doing simulation studies. We're looking at further application development to other target properties, as I mentioned, such as energy content, reproduction. We're also looking at field instruments and needed labor to acquire data, measure successes. We may have to advance skill sets and training requirements to support model implementation, which we are already looking at. And then finally, in the operationalization phase, how to best communicate a better understanding and an adoption of this new aging paradigm to the stock assessment users, as well as the management organization. So with that, I would be glad to answer any questions. And I apologize for cramming all that in such a short space, but we're doing a lot. Thank you.
All right, thanks a lot, Tom. I've got right now roughly 7 questions, and there may be some more coming in as you answer some, but folks are welcome to enter their questions via the question tab on the right-hand side of their webinar control bars. But jumping right in, and some of these are a little bit overlapping, and they almost all focus on otolith questions. But the first one comes from Dr. Timothy Lohr, and says, you've suggested that the primary age-dependent change in absorbance profiles is related to nitrogen-carbon dynamics, which suggests some manner of age-dependent shift in the otolith matrix. Is there any evidence supporting hypotheses regarding the molecular nature of that shift? For example, fundamental changes in either protein to amino composition of molecular conformation amongst protein units, or changes in the relative proportion of protein to carbonate, or the manner, for instance, binding regions in which the carbonate binds?
Tim, that's a great question. Certainly a mouthful, and talks to a lot of the biochemistry that might be involved in the actual molecular variants of protein and other constituents in the otolith. One of our collaborators, Oliver Thomas from Australia, University of Melbourne, has published a good number of papers on the proteomics of otoliths. He's identified up to 300 different specific protein structures in variants, some of which have a very important functional role in the building of calcium carbonate or the aragonite structure of the otolith itself. The question you're asking, I think in theory has a fundamental basis, as we've been talking to Thomas or Oliver Thomas about this. But that certainly is a big topic that we are pursuing at the moment, because we have hired Oliver Thomas on a postdoc to actually help us extract and identify some of the molecular constituents, or the proteins specifically, that are related specifically to the wave number information that we're getting from the spectra. I know that's not exactly the answer you want to hear in terms of what I know, but it's basically what we're doing. And I'd say that's the answers are yet to come.
Sounds good. Thank you. Okay, next question, also from Dr. Lohr, is regarding species and ecosystem signatures. And he's wondering if you have any idea on how long these ecosystem signatures would remain conserved. Specifically, would the signatures be stable in otoliths from historical middens and geological deposits? Either cultural or climate reconstruction ideas there.
Good question again. I would say from the fresh otoliths, there would be strong conservation in terms of the target molecular properties that would remain in the otolith. From middens, it depends on the amount of diagenesis that occurs with the weathering and leaching. What we have seen in our otoliths that we've analyzed from middens, and in one study we're looking at a midden that we've collected Pacific cod otoliths that go back 6,000 years, is that in many of those cases the aragonite structure is turned to calcite or veterite, some other form of more stable bicarbonate. And my guess is that because of that, it's going to cause a complete deterioration of protein, if in fact the protein is responsible for the spectral information. My guess would be probably not conserved in archaeological specimens, but you never know.
Okay, sounds good. And that leads partly into the next question. I'm going to probably pair these two questions up. The first one is from Andrew Chin, and it is asking whether you have tested vaterite otoliths. And then the second one is kind of related on the topics of logistics with otoliths that you're testing, comes from Dana Rudy. And it is, does glycerin interfere with FT-NIRS reading? How about the curvature of the otolith? Are there any deal breakers for using the FT-NIRS technique or species that you've run into trouble with?
I'll start with the last question first. I would say of all the species we've analyzed, and let's specifically talk about species where we say analyze more than 1,000 otoliths. The list is not huge. It's probably close to a half a dozen, and in each of those instances, I do not see a operational problem with regard to the FT-NIRS with regard to age estimation. In some cases, I see huge advancements in terms of the efficiency gains, particularly in some of the longer-lived rockfish, where I think the time it takes to process and actually age the otolith could be upwards of, say, 8-9 minutes per otolith. Whereas, basically once we've got a calibrated model, the time it takes to generate an age prediction from FT-NIRS is only basically 30 to 45 seconds. So I don't see any particular problems, but then again, I would say that we may indeed see problems with other species that may have inherently problematic patterns in the otolith. One thing we have to keep in mind is that in order to develop calibration and validation and to validate that model, we have to pair the traditional age information up with the spectral information. So to the extent that the traditional age information is horrible, that is going to basically be problematic for the age estimation from FT-NIRS as well. So like I say, in all cases, it's not a technology that will be broadly extended. An example would be short spine thorny head, which is a relatively long-lived species and for which we see CVs on the order of 15% or percent agreements plus or minus 0 years down to 13%. So yeah, there will be obviously, cases where it doesn't apply well. The other topic related to that is that what we're trying to do, because we recognize that the reference data that we're using, which is the traditional age data, is biased, or maybe not biased, but certainly imprecise, or it has uncertainty. And so one of the techniques we're looking to explore is actually an age-specific bias correction in the prediction or in the calibration model itself, which could reduce bias from a subset of data that have been truly validated by some other methods. To go back to one of your other questions of vaterite, we have not done, let me just say that we've scanned some otoliths that have what appears to be significantly more vaterite or some form or more stable form of bicarbonate, such as calcite, and that potentially may be problematic. We have not fully investigated those circumstances. One of the reasons is because a lot of the species that we analyze, we don't see too much diagenesis in the otolith. The question whether thymol has a fingerprint that is interferent, so to speak, in the spectral information. Yes, we've looked at the spectra of thymol, and we can identify that by simply analyzing thymol by itself and also removing it from the spectra that we get from otoliths using some transfer techniques. And then the last question deals with curvature and funky shapes. Those do in fact have the potential to affect the spectra. The reason is because based on Lambert-Beer's law, one of the critical parameters in the detection of light has to do with the path length of the energy. And path length can be affected if the otolith, for instance, has a great deal of curvature and is put on the sample window or the scan window when it's concave down, so that what you're essentially doing is increasing the path length from the source to the sulcus, or the point of the otolith where there's got a good deal of curvature. And what we've done early on is we've analyzed different presentation modes to evaluate what is the best and most reliable way to present the otolith on the sample window. Generally, what we have done is actually presented the otoliths with the concave portion up rather than down. And that would essentially control to some extent for differences in path length if there's a great deal of curvature.
Great, thank you. Next, we have two questions from Jeff Tingley, and I'll just read them both out. The first is, and you touched on this on one of your previous questions, but how is this approach likely to respond in a situation where traditional aging was systematically incorrect? For instance, where traditional aging shows a substantial under or overestimate for all the fish aged. And then secondly, once this approach is established, what would the typical cost per otolith to determine age be?
Great questions. As I said before, any predictive model will be as bad or sort of influenced by any bias that may exist in the traditional age data. As we all know, though, at least from a stock assessment perspective, is even if there's bias, it can be corrected for internally in the stock assessment. If, say, an aging bias vector, say, were to be incorporated in association with an age age precision matrix or an age precision vector in a stock assessment. My preferred way would be to try to correct for the bias in the new model development process. And again, in both cases, it would require a separate study for which you've actually conducted an analysis and quantified the age bias that's associated with each age in the stock assessment. But either way, it can be handled. So I don't view that as a problem. Again, because largely there's going to be a one-for-one substitution of the traditional age by the new FT-NIRS age. And whether it's basically corrected externally from the stock assessment in the calibration validation process, or whether it's corrected for in the stock assessment, should handle each of those scenarios. I forgot the other question, Ian. Could you repeat it, please?
So the last question was about your kind of estimate of costs once you have
a
model set up and running. Is there some sort of estimate therein?
Boy, I wish I had that information at this point. You know, I could say it's substantially less, and I don't really have the numbers to support that. It's based on the fact that, you know, you can buy an instrument. Well, let's back up a minute and just kind of recognize the fact that the goal of the technology is not to replace microscopic age readers. So in the long term, it may in fact reduce the number of people engaged in microscopic aging. The fact is that moving forward in our operational process, you're still going to have to update the model, and you're still going to require some traditional age estimation using microscopic evaluation. That's simply because you still want to, number one, enhance or support the expertise that you have in-house to actually, you know, estimate age from that method, but also to update a model, it requires paired data of both microscopic age and the FT-NIR spectrum. So one thing is for certain is that under an operationalized setting, you probably wouldn't need that many age readers. As far as cost goes, one for one, an instrument may, you know, the cheapest instrument, for instance, the Tango, costs roughly, I think, $65,000. And it's a robust instrument. It has, you know, expendables that are relatively modest in price, such as new light sources, a new laser, desiccant, you know, desiccants and that sort of thing. And certainly it would fall within the budget of a typical production aging laboratory. But if you compared, say, just on a case-by-case basis of the instrument versus, say, the amount of money it takes to support an FTE, there's a very large differential there. But again, you know, I think if you actually put it into a cost function where you're trying to identify expenditures and revenues, you know, or some other measure of benefit, you know, you would have to take into account, you know, the amount of time that's being required to process a given otolith and the number of FTEs that are required to process such an otolith. For the species that we've analyzed, and these are some of the easier species that require less analysis time, walleye pollock, for instance, requires on average about 3 to 5 minutes per otolith over all age groups from a single analyst. Whereas, you know, if you had a developed quantitative model or a quant model that the instrument is running off of, you're looking at basically a difference of between 35 and 45 seconds. So that's an order of magnitude difference in terms of time. And therefore, if you related time to time on the basis of, say, you know, prorating how much time, the instrument life with that time, and prorating, you know, your aging expert with the time required, you know, per unit, I think you would see a very large differential again in terms of the cost savings. I think I need to talk to our economists in AFSC to try to put this into a utility function and try to really answer that question in a more quantitative way. We haven't done that, but I think more or less the basic facts kind of speak for themselves.
All right, Tom, I want to thank you for an outstanding presentation and for being willing to stick around and answer questions for a pretty long amount of time here. I know that we still have a few questions outstanding. Would you be willing to field questions via email if people want to follow up after this presentation?
Yes, very much so. And if people are interested in looking at, you know, our 100-page document from the proceedings of our first workshop, that is on the NOAA website. And there's a number of other links on the NOAA website to the published papers that talk about these applications. So, but if anybody wants you know, a PDF, or if they want a link, they can just respond to me via email, and I would be glad to answer any questions via email as well.
All right, thanks, Tom. And that's Thomas Helser at NOAA.gov.
Yeah, you bet.
All right, and I want to thank everybody for sticking around. I see we've still got a pretty high attendance level. I note that we were at least twice our room capacity, so there's some silver lining to having to do these meetings in this format, because you got quite a broad reach with this presentation. Before we go, I do want to invite everybody to join us next week, the 17th of February, for our second seminar in the 2021 series, which will be Lauren Wild, changing it up completely and talking about whale depredation. So thanks again, everybody, for joining us, and thanks, Tom, for a great presentation.
Yeah, many thanks for having me. Appreciate it.
All right, goodbye, everybody.
Goodbye.